Welcome to Science Store online classes.
Don't forget to like our video and subscribe our YouTube channel for all the updates.
Welcome to all my dear students. Today I am taking biology class. Under that we are studying the chapter Plant Kingdom. In last class we have studied about the general characters of Gymnosperms as well as Angiosperms, how sexual reproduction takes place in Gymnosperm and the general characters of the Angiosperms. Usually we have studied the different, this Angiosperm contain two main classes, that is dicot and monocot. Okay, dicotyledons and monocotyledons. And for example, good example for dicot plant is your hibiscus or your flowering plants, garden plants which flowers well, that are all example for your dicotyledon type of plants. Even mustard, any of the brinjal and even lady's finger, these are the example for angiospermic dicotyledonous plant. And grass, rice, wheat, grass, these are the example for monocot plants. Okay, next we have studied based on the size, these are different divided into trees, shrubs and herbs. And also you can say smallest angiosperm, that is Wolffia, and tallest angiosperm is eucalyptus, which measures up to more than 100 meters. Okay, and we have studied the reproductive structure. Usually in angiosperm, reproductive structure is developed in a specialized organ called flowers. Flowers contain male and female reproductive organs, that is anther and stamens, and this stamens and pistils are the two important male and female reproductive structure of the angiospermic plants. Here it is also called as Androecium and Gynoshem, no need, but you should know Androecium means male reproductive structure, male sex organ is also called as Androecium, female sex organ is also called as Gynoshem. Gynoecium. Okay, female reproductive structure is called as Gynoecium and male reproductive structure is called as Androecium. That is not needed. Okay, next we'll move to the structure, how the stamens look like. I have explained the structure of stamens, which contain a long filament like structure called filaments and lobed structure. One at the tip you can see the lobed structure, that is nothing but the anther. Inside the anther, okay, inside this anther, if you take a cross section of the anther, it looks like this, looks like a butterfly. Okay, you are going to study about this in detail in your second year classes. Okay, second year classes alli structure ಬಗ್ಗೆ detail ಆಗಿ study ಮಾಡ್ತೀವಿ. This is a cross section of the anther. If you take it this like this, if you take a section like this, you will get like this structure. Okay, here you can see the sac like structure which bears a small minute spores developed from the small minute spores called microspores, developed from pollen mother cell. Here some of the anther cell differentiate into pollen mother cell. They differentiate and form a pollen mother cell, and this pollen mother cell undergo meiotic division and forms microspores. Here ಏನಾಗ್ತದೆ ಇಲ್ಲಿ ಕೆಲವೊಂದು cells, the cells of the anther which undergo meiotic division and forms a microspores. Okay, this usually that cell which undergo meiotic division is called as pollen mother cell, and that meiotic division give rise to the formation of microspores. Here these microspores These microspores later differentiated or later develop into pollen grains. That is male gametophyte. It is nothing but a male gametophyte. This is nothing but this pollen grain is nothing but a male gametophyte which is produced from microspores. Here microspores undergo further division and forms pollen grains. It is nothing but a male gametophyte. Same way in Gynoshem, I have drawn the structure of Gynoshem. At the base you can see swollen base. At the tip you can see the stigma. This is stigma. This is style, and this part outer along with the ovule is called as ovary. This part is ovary, it contains ovule. Okay, this contains ovule. This ovary, inside the ovary you can see the smaller round spore like structure, that is nothing but the ovule. Okay, this ovule which is developed from the megaspore mother cell. Here it is usually the ovary, the cell of the ovary forms a megaspore mother cell. Mother cell which give rise to the formation of megaspores. ಏನಾಗ್ತದೆ, ಮೆಗಾಸ್ಪೋರ್ ಈ ಓವರಿಯಲ್ಲಿ ಕೆಲವೊಂದು ಸೆಲ್ಸ್ ಮೆಗಾಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಆಗಿ ಡೆವಲಪ್ ಆಗ್ತದೆ. ದಿ ಡೆವಲಪ್ ಇಂಟು ಮೆಗಾಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಅಂಡ್ ದಿಸ್ ಮೆಗಾಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಅಂಡರ್ಗೋ ಮಿಯೋಟಿಕ್ division, that is reduction division, and forms the megaspores. Here the megaspores, 4 megaspores are formed from each megaspore mother cell. Here out of 4 megaspore mother cell, megaspores are formed from single megaspore mother cell. This is megaspores formed from this megaspore mother cell. 4 cells, megaspores will be formed. Okay, here out of 4, 3 will degenerate. Here out of 4, 3 will degenerate. 3 megaspore will degenerate and single megaspore develop into a ovule. Okay, the single mother cell develop into a female gametophyte. Okay, here this develop into a female gametophyte. Okay, here this female gametophyte is 3 or egg apparatus, egg sac that develop. The female gametophyte is nothing but egg sac. The sac contains egg cells. The sac which contains the egg, andre sac andre somewhat bag like structure anna sac heltare. So that sac, egg containing sac like structure, will call it as egg sac, which is also called as female gametophyte. Here in this female gametophyte you can see this female gametophyte or egg sac is 3 egg having a 3 egg apparatus. This is a structure of, it is somewhat oval in shape. You can see somewhat oval in shape. This is divided into 3 egg apparatus. You can see inside this egg sac you can see 3 egg apparatus. It is divided into, partially divided into 3 parts. Okay, each part 3 we'll call it as egg apparatus. Each section will call it as egg apparatus. Each section contain different kinds of the cells. I am going to explain. Ee 3 section, each egg sac untalla 3 section agi divide agtade. That we will call it as egg apparatus. Each egg apparatus contain different kinds of the cell. Here at the one side, at the base you can see one large egg cell. Here you can see one large egg cell. Okay, here this is a large egg cell. This is egg. This is egg cell, and beside this you can see smaller further they divide and form. Here this megaspores, one megaspore will form into egg female gametophyte. That for the formation of female gametophyte they undergo frequent division ಆಗ್ತದೆ. They form about 8 cell structure develop ಆಗ್ತದೆ. Here it contains on the 2 sides of the egg cell you can see the cells called synergids. The 2 cells present beside the egg cell is called as synergids or synergid cells. Okay, on anterior end, this is posterior end. This egg is developed in the posterior end. In the anterior end, you can see 3 cells. Okay, 3 cells. These cells we will call it as antipodal cells. This anterior end it is having a 3-celled structure, that we will call it as antipodal cells. This entire structure is a egg sac. Okay, each section we will call it as egg apparatus. On one apparatus it contains antipodal cell, 3-celled structure we will call it as antipodal cells. At the base you can see the egg, presence of egg with the 2 synergid cells. Okay, at the center you can see the development of polar nuclei. Here all these cells are haploid in structure. All these cells are haploid in nature. Haploid in nature. Okay. Haploid in nature, and these central nucleus and central cells are called as polar nucleus. Polar cells. Later what happens, these polar cells will fuse, it will fuse and form a diploid structure. It form a diploid cell, single cell. It fuse and form a single cell. We will call it as polar nuclei. Because it contains 2 nucleus ಅನ್ನು ಹೊಂದಿರ್ತದೆ. The plural of nucleus is nuclei. Okay, remember the plural of nucleus is nuclei. Here this polar nucleus will fuse and form a single structure, diploid structure, that will call it as polar nuclei or polar cells. Polar cells anta heltave. This is a structure of embryo sac or female gametophyte. This is a complete structure of female gametophyte. Outerly it may be covered with a thick fleshy part. Okay, it may be outerly covered by outer part, that is ovary. This outer covering we'll call it as ovary, and this entire structure which is inside this structure, inside this you can see a ovules, which ovule contains this embryo sac. Embryo sac contain different kinds of the cell. Okay, this is about the structure of ovules or embryo sac. Inside the ovule the structure looks like this. Female gametophyte hige develop ಆಗ್ತದೆ. Okay, next we'll move what happens, this pollen, how the fertilization takes place. What happens, this pollen anther which give rise to pollen grains, right alva? Pollen grains anna adu produce ಮಾಡ್ತದೆ. I have mentioned pollen grains. That pollen grains, this megaspore will further and divide, form a male gametophyte called pollen grains. Okay, like this thick coating develop ಆಗ್ತದೆ, because it have to fly freely in the air. So it should have a resistant covering ಇರಬೇಕು. So it form a spine like structure, outer cover, and it may contain 3 or 4 cells. 3-celled structure ಆಗಿ ಡೆವಲಪ್ ಆಗಿರುತ್ತೆ. ಇದನ್ನು ನಾವು pollen grains ಅಂತ ಹೇಳ್ತೇವೆ ಅಥವಾ male gametophyte ಅಂತ ಹೇಳ್ತೇವೆ. So what happens, after complete maturation the pollen grains are released from the anther or dispersed from the anther. Okay, it is dispersed or it is transferred to the male gametophyte or the male reproductive structure in the air. It will open. This it forms this anther forms a small minute microscopic opening that through this opening the pollen grain get released into the outer environment. It may be carried by air currents or waters. The pollen grains may be transferred by various means, by wind, by air, or by the insects. Some of the insect which go and sits on the flower for nectar purpose ಅಲ್ವಾ, ನೆಕ್ಟರಿಗಾಗಿ ಅದು ಫ್ಲವರ್ ಅನ್ನು ಡಿಪೆಂಡ್ ಆಗಿರ್ತದೆ. when it goes to the flower, the pollen grains will stick on to the surface of the body. When it goes to another flower, this pollen grain will be dusted on the stigma. Okay, another flower idu stigma mele dust agtade. So by this way pollination, the pollen grains will be transferred to the stigma, get transferred to the stigma of the another flower or same flower ಅಥವಾ ಬೇರೆ ಫ್ಲವರಿಗೆ ಹೋಗಿ ಸ್ಟಿಗ್ಮಾ ಹೋಗಿ ಕುತ್ಕೊಳ್ತದೆ. The process of transfer of pollen grains from anther to stigma, that process will call it as pollination. What is pollination? Pollination means the process of transfer of pollen grains from anther to the stigma. That process we'll call it as pollination. Pollination may be of different types based on the mediator or based on the carrier. You can see different kinds of the pollination. It may be through insect. If it is from insects, it is a zoospore. Different names are there depending on the organism involved in the pollination. Pollination ಗೆ ಇನ್ವಾಲ್ವ್ ಆದ ಯಾವುದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಇನ್ವಾಲ್ವ್ ಆಗುತ್ತೆ, ಯಾವುದು ಮೀಡಿಯಾ ಇನ್ವಾಲ್ವ್ ಆಗುತ್ತೆ, ಅದರ ಡಿಪೆಂಡ್ಸ್ ಅಲ್ಲಿ there are different types of pollination. Okay, that you are going to study in later classes or in second year classes. ಅಲ್ಲಿ second year classes ಅಲ್ಲಿ ಕಲಿತೀರಿ. Okay, what happens? Okay, I'll draw here itself. Okay, this form a small style. This is style, right? Idu ovary wall. This is ovary wall. This is style, and at the tip you can see the stigma. You can see the stigma. Here pollen grains will fall on the surface of the stigma. Okay, when it falls, it start to produce the pollen tube. Okay, it start to produce the pollen tube. This pollen tube, this germination of the pollen grain, the tube formed due to the germination of the pollen grain is called as pollen tube. This pollen tube will carry the male gametes. It will carry the male gametes downwards. Okay, towards it carries the male gametes into the cell. Okay, like this. ಇಲ್ಲಿವರೆಗೆ ಅದು pollen tube germinate ಆಗ್ತದೆ till it reaches the ovule. egg cell ಗೆ reach ಆಗುವವರೆಗೆ this pollen tube will be formed. Okay, ಇಲ್ಲಿ ತನಕ ಬರ್ತದೆ. Next later it will move to the here. Okay, and it fuses with the egg cell. Okay, it fuses with the egg cell and form a zygote. The fusion of the male gamete, with female gamete, the process of fusion of male gamete with female gamete, this process we'll call it as syngamy. The process of fusion of male gamete with the female gamete, this process will call it, this fusion will call it as syngamy. Okay, very important, syngamy. Here syngamy takes place. The male gamete will fuses with the egg nucleus and forms the zygote structure. Later it form the zygote structure. This is a structure of the zygote, and one more process takes place here. This pollen tube, another male gamete will fuses with the polar nuclei, diploid polar nuclei. Here it fuses with the diploid polar nuclei and forms a triploid cells. It forms a triploid cells. Okay, here the one of the male gamete fuses with the polar nuclei and form a triploid cell. This cell will call it as primary endosperm nucleus. This is nothing but PEN, primary endosperm nuclei. That when the another male gamete when fuses with the polar nuclei and form a triploid structure, triploid cell, this triploid cell will call it as primary endosperm nucleus. This triploid structure anna primary endosperm nucleus anta mention ಮಾಡ್ತಾರೆ, and this will call it as egg. This will forms a zygote. Okay, this forms a zygote and this forms a primary endosperm nucleus. So this the fertilization, the process of fusion of gametes, male gametes with female gametes, will call it as a fertilization. Here the two fertilization takes place. One of the gamete fuses with egg, another fertilization with polar nuclei. Two fertilization takes place. This process will call it as double fertilization. This process of fertilization, two fertilization taking place at a time. This process will call it as double fertilization, which is very important, seen only in angiospermic plant where there is a development of primary endosperm nuclei and zygote. There is at a time you can see the formation of primary endosperm nucleus as well as zygote. Okay, two cells are formed at a time. Okay, so that fertilization will call it as double fertilization. Here the zygote will give rise to the formation of seed. It will give rise to the formation of seed, and here the primary endosperm nucleus will develop into a nutritive substance to the embryo. Sorry, this will develop into the zygote develop into embryo. The zygote develop, they divide and form a embryo. Here this primary endosperm nucleus develop into a endosperm. They undergo further division and form a endosperm around the embryo around endosperm agi form ಆಗ್ತದೆ. This endosperms are nothing but the nutritive material present in the seed, nutritive content of the seed which nourishes the embryo. While embryo develop ಮಾಡುವಾಗ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗುವಾಗ, ಪ್ಲಾಂಟ್ ಆಗಿ ಡೆವಲಪ್ ಆಗುವಾಗ ಅದಕ್ಕೆ ಫುಡ್ ಬೇಕಿರ್ತದೆ, ನ್ಯೂಟ್ರಿಷನ್ ಬೇಕಿರ್ತದೆ. ದ್ಯಾಟ್ ಈಸ್ ಸಪ್ಲೈಡ್ ಬೈ ದ ಎಂಡೋಸ್ಪರ್ಮ್. ಓಕೆ, ದಟ್ ಈಸ್ ಸಪ್ಲೈಡ್ ಬೈ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಎಂಡೋಸ್ಪರ್ಮ್ ಅರೌಂಡ್ ಇರ್ತದಲ್ಲ, ಅದು ನ್ಯೂಟ್ರಿಟಿವ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಅನ್ನು ಪ್ರೊವೈಡ್ ಮಾಡ್ತದೆ ಎಂಬ್ರಿಯೋಗೆ. ಓಕೆ, ಯೂಶಲಿ ಅ ಗುಡ್ ಎಕ್ಸಾಂಪಲ್ ಆಫ್ endosperm is your coconut. You are using coconut, no? That white color part inside the hard shell, you can inside that you will get one white colored part. Okay, coconut. Okay, that is nothing but the endosperm. We are consuming it. Most of the endosperm is very nutritive in nature, so very good for health, good for the health. So these are used for even animals also use this endosperm for their nourishment. And here usually these endosperms are developed to nourish the embryo. Ellinda endosperm formation agutte, and all these endosperms are triploid in condition. Ella cells galu ee cell inda adantaha ella cell galu triploid condition alli irtade. Mooru set of triploid means 3 set of chromosome containing cells. Okay, triploid condition means 3 sets of chromosome. Here this endosperm will be formed from primary endosperm nucleus. Okay, understood? Okay, this is the importance of the process of double fertilization. Okay. Okay, next will this embryo will later, here this all takes place inside idre olage agudrinda this embryo sac later develop into seed. Okay, embryo sac later develop after fertilization, after complete maturation of this embryo and endosperm formation. It forms a seed structure, and around this you can see the wall, right? Idanenu helidane ovary. This is nothing but the ovary. This wall later develop into a fruit. Here ovule develop into seed and ovary develop into fruit. The fleshy part around the fleshy part will consume, no, that is nothing but the fruit part. Okay, and here inside if you take a mango nodidiralla, you have eaten, all of you seen mango fruit, and also you have tasted the mango fruit. Here usually the mango is like this. Here you can see centrally, you can see the seed. This is seed and this is fruit. This developed from the ovule and this developed from the ovary part of the flower. Ovary part irtadalla, pistil idu ovary part fruit ಆಗ್ತದೆ, ovule part seed ಆಗ್ತದೆ. Okay, very important. And usually why these angiosperms are useful, you know, or most of the angiospermic plants are very useful for animals as well as human beings, alva? In which and all way we can use the angiospermic plant. For what purpose we can use the angiospermic plant? We use it for food, shelter, right? Most of the angiospermic plant, this monocots, cereals, millets ಎಲ್ಲ produce ಆಗ್ತದೆ by the monocot plant. Even pulses, various types of the seeds which is developed from the angiospermic dicot plants that are used for the food purpose. Even potato, what will eat, vegetables, which are all from the this angiospermic flowering plant will give these products. So food purpose is used for food purpose, and shelter. Most of the timber yielding plant are used to construct the houses. In ancient days or in olden days, this most of the tall trees or the timber, the bark or the stem part is used for the construction of the houses. Next, many clothing, many fiber yielding angiosperm, fiber yielding plants which is used to prepare the cloths. Textile industry, very important for textile industry. For example, cotton is nothing but cotton is a plant origin. It is formed from the fruit, around fruit. Okay, this is a seed around. You can see the development of fruit part. Okay, this fruit part in the cotton plant is nothing but a cellulose fiber. It is nothing but this fruit part is developed from the cellulose fiber. After maturation, after it dries up, these fibers are clearly separated. Okay, initial stages alli adu compact agi arrange agirtade. After maturation, these cellular fibers are dries up and form filaments. Okay, that is used for the cloth making, and also some of the jute is also used in preparation of the some cloths. Okay, cloth. Even linen also plant origin. Linen is a type of cloth material which is originated from the plants. Okay, these are very important. Next, most of the plants are medicinally useful. Very important. Most of the Ayurvedic medicines are extracted from, and they are from plant origin. Most of the Ayurvedic medicines, ನಾವು ಯಾವುದು ಆಯುರ್ವೇದಿಕ್ ಕಷಾಯಗಳನ್ನೆಲ್ಲ ಕುಡಿತೀವಲ್ಲ, we consume that kashayas and even Ayurvedic tablets. These are nothing but the plant extracts ಅನ್ನ ಮೆಡಿಸಿನ್ ಆಗಿ ಕೊಡ್ತಾರೆ. And also it is used for various purpose, like other purpose like paint preparation, natural organic paint prepare ಮಾಡಲಿಕ್ಕೆ. Then this gum for the extraction of various plants are gum yielding. From that they will extract the gum and used for various purpose, and also different uses are there, plant uses. Can list out any uses of the angiospermic plant. Okay. Okay, these are the different uses of the plants. Next we will move to the life cycle. We will study the life cycle of the plant. Okay, draw down the structure of the life cycle of the plant. Here first plant will be formed, alva, with the root system, well developed root system. The plants have well developed root system. Stem, here angiospermic plants have well developed root system, stem system and leaf system. Here this will give rise to flowers. This is the structure of flower. Okay, flower which having a anther. Okay, it is having a anther. And the structure, and it is also having female reproductive structure, that is pistil. This is stamen and this is pistil, which contain ovary. Okay, and this will give rise to the microspores, and this will give rise to the megaspore. Okay, later this develop into pollen grains. Okay, here it will develop into pollen grains, that is male gametophyte. Okay, this develop into embryo sac with one egg cell, 2 synergid, 2 polar nuclei and 3 antipodal cell. This is a embryo sac structure, and it this germinate this pollen grain germinate and form a pollen tube. Okay, that lead to the fertilization, that lead to the formation of zygote. Okay, that lead to the formation of zygote. Zygote later develop into embryo. Embryo usually like this structure, heart shaped structure iruttade usually embryo. So this embryo zygote, this embryo later develop into they divide further and form a 2 cotyledon structure. Cotyledons develop agtade. Usually the endosperm in monocots you cannot see this cotyledon, single cotyledon is present, and also that is not nutritive in nature. But here in dicot you can see 2 well developed cotyledons which are they lack endosperm. Okay, dicot alli endosperm lack agirtade because the nutritive supply is done by the cotyledons. Here you can see the development of cotyledons from embryo. Embryo give rise to cotyledons. Okay, usually the cotyledons are developed from embryo part of the embryo part in the cotyledon develop agtade. So this is a structure of, this is multicellular structure, embryo is multicellular structure. Okay, they may be present with 2 cotyledon structure. ಈ ಟು ಕಾಟಿಲಿಡನ್ಸ್ ಮಧ್ಯೆ ಈ ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ ಆಗ್ತದೆ. ಓಕೆ, ಆ್ಯಂಡ್ ದಿಸ್ ಎಂಬ್ರಿಯೋ ಲೇಟರ್ ಗಿವ್ ರೈಸ್ ಟು ದ ಫಾರ್ಮೇಷನ್ ಆಫ್ ದ ನ್ಯೂ ಪ್ಲಾಂಟ್. ದಿಸ್ ಕಾಟಿಲಿಡನ್ or the embryo will germinate and form a new plant. This cycle continues. Okay. Here, which are the diploid structure? This nothing but sporophyte. The plant, the actual plant body is nothing but the sporophyte. This is anther. This is pistil. Okay, this is microspore. This is megaspore, which is also called as megaspore mother cell, which is developed from megaspore mother cell. This is embryo sac. This is pollen grain. This is pollen tube formation. This lead to the formation of zygote. This is embryo. This embryo later develop into here this is haploid in nature. This is diploid. Okay, this is diploid. This is haploid. This is diploid. Again the zygote become diploid condition, and it is also diploid condition. You can see maximum diploid condition only it is confined to the here this gametophyte formation. Only in gametophyte formation you can see the haploid condition. Rest of the condition will be diploid in nature. Rest of the stages contain diploid type of the cells. Okay, this is a life cycle of the angiosperm. Okay, very important, this double fertilization and PNE, that is primary endosperm nucleus, structure of the embryo sac, how the polar nucleus, how triploid cells are formed. Okay, then next the triploid cell will later develop into endosperms. Here zygote later develop into embryos. Okay, embryo develop ಆಗ್ತದೆ. Next the entire embryo structure, the ovule structure develop into seed, and entire ovary structure will develop into fruit part. Okay, this will degenerate, stigma ಮತ್ತೆ style part degenerate ಆಗ್ತದೆ. So after fertilization this egg ಮತ್ತೆ polar nuclei fertilize ಆಗ್ತದಲ್ಲ, the remaining synergid and antipodal cell will degenerate. After fertilization of egg and polar nuclei this will degenerate. ಇದು degenerate ಆಗುತ್ತೆ, gradually degenerate ಆಗ್ತದೆ. So because it is not having any work after the fertilization, so they degenerate. Only this fertilized egg and fertilized polar nuclei will remain, and later they after fusion syngamy they later convert into next stage. Okay, this is the life cycle and the important points of the angiosperms. Okay, angiosperms are the important points, and also these angiospermic plants are very useful and most extensively present. You can see these angiosperms in any habitat. The habitat in aquatic form you can see in saline condition, marine condition alisa can see some in beach siders you can see some angiospermic plant, and also you can see in dense that desert condition alisa can see the development of some angiospermic plant. Even in the cold condition, even in extreme temperature, less humid condition, you can see the development of angiospermic plant, and it is very important. So most of the angiosperms are used as a ornamental plant, decoration. Other uses of angiospermic plant is decoration, and they may be used in gardens for the decorative purpose, and stage decoration allige use ಮಾಡ್ತಾರೆ angiospermic flower anna. So this angiospermics is also called as flowering plant. It is also called as flowering plant, where you can see flowers only in this division. Okay, in angiospermic division alli matra you can see the development of beautiful flowers. Remember, rest of the plant kingdom will not flower. This angiosperm only will flower, and this flower contain the reproductive structure, specialized structure meant for the reproduction. Flowers are nothing but the specialized structure meant for reproduction purpose. Okay. Thank you all my dear students. Will continue with the next that life cycles, different life cycles. Okay.
Don't forget to like our video and subscribe our YouTube channel for all the updates.